So, hello everyone. Thank you a lot for joining us today for today's webinar. We will have Professor Luca Fontanesi. He's a full professor of livestock systems and animal breeding and genetics, and he will give us today's presentation about environmental DNA and multiple kingdom derived fingerprint for honey. Professor Luca, the floor is all yours.
Thank you very much for giving me the opportunity to present this topic today. Good afternoon, everybody. Thank you for being here today. I will present what we did over throughout the last few years, working on environmental DNA, mainly on honey. Just a few words before starting. I'm from University of Bologna. Bologna is a quite known town in the north of Italy, and is a unique town preserved from the Middle Age period. And University of Bologna is the oldest university in the Western countries. It was founded in the year 1088, and we are quite proud of that. So University of Bologna is spread all over the region, and we have also campus in Buenos Aires. I'm coming from Department of Agriculture and Food Sciences, which is one of the largest departments of University of Bologna. It is divided in 10 divisions, and I'm involved in the animal science division. I'm leading the animal and food genomics group at the University of Bologna, and here are the people that contributed a lot to what I will tell you today. And most job has been done by Samuele, Anisa, Giuseppina, Matteo, Valeria, and especially so other people that came with us for a period of training and fellowship. Dalal, Valerio contributed a lot, Kate, Aisha contributed a lot, and also Francesca. Just a few words of what we are doing, animal and food genomics. So we are working mainly in livestock genomics, most of the livestock species, aquaculture species, and also other species, and also in food genomics, pig meat, dairy, and honey, and other type of food, processed foods using genomics. But of course, the bridge between the animal and food is honey, and for this, we are investigating apicultural genomics topics, including also Vespa velutina, Etina tumida, and also Varroa destructor, and other topics. Okay, just after this brief introduction, we can go back to the main topic of this presentation, environmental DNA. What is environmental DNA? It is DNA that is collected from a variety of environmental samples rather than directly from an individual organism. So we cannot say that honey has its DNA. Its DNA comes from all organisms in some way that have been directly or indirectly in contact with honey. So through the production steps, all production steps, from the beginning, the nectar harvesting from the honeybees to the processing that honeybee do, conservation, and then also transportation and so on. So there is a interesting fingerprinting that is left by all organisms that in some way left the DNA into the honey. Honey is also very simple to sample. Of course, you can go to the supermarket and buy honey samples and get some information from that. And there is a very peculiar element in honey because commercial honey, or maybe just a bottle of honey, contain honey that is produced by multi-colonies. Of course, one colony contains 1,000 of honeybees, so the contribution is quite unique. So we can investigate at different levels the environmental DNA that derive from all these elements. It is useful for what? Environmental DNA contained in honey. We can use environmental DNA contained in honey as a tool to do several activities that could also contribute to combat frauds. There is a possibility to authenticate honey in some way. And here I listed just a few elements that can be in some way targeted to obtain this type of information, botanical, entomological region. It is possible in some way indirectly to authenticate honey describing this kingdom to obtain geographical information. It is possible to use environmental DNA for also detecting or obtaining information to describe population genetics of the honey, and not only on the honey. And this could be also important for conservation genetics to measure inbreeding level of the colony and so on, and also to obtain information on the health of the colony. It could be possible to use it as a monitoring and epidemiological tool. We can get information from pathogens, parasites, pests, and so on. And of course, finally, we can combine all this information to obtain a sort of hologenomic information derived from this food matrix. What we did over throughout the last years. So we collected a few thousand honey samples. At present, we have approximately 10,000 honey samples collected in our biobank, honey bank, derived from different sources. We have isolated DNA from all honey samples, almost all honey samples, and then we have analyzed the DNA that we isolated in several different ways. We used two approaches. One can be considered a target approach. They could be divided in two types of approaches. Endpoint PCR analysis, just to get information on a very specific target and analyze the presence or absence of some DNA fragments. And then so we use target analysis combining next generation sequencing in metabarcoding approaches. We also used on-target analysis. So we isolated the whole DNA from the honey, and we next generation sequenced what we obtained. This approach is called sometimes shotgun metagenomic analysis, whole DNA sequencing, and so on, using next generation sequencing platforms. All right, so I will start focusing on target analysis for different approaches using the combination of the endpoint PCR analysis and next generation sequencing in metabarcoding analysis. I will discuss first elements that could be useful to combat frauds, plant DNA, plant stacking instant DNA, and then we'll move on genetic diversity aspects and then also health aspects. All right, the first element, plant DNA, how we can in some way use plant DNA to obtain information about the geographical origin of the honey that could indirectly be useful to authenticate it. We published a paper a few years ago where we described the approach. Of course, the honey contains pollen that is carried out by the bees during the jobs, and this pollen is from plants, and we have pollen into the honey. Of course, melissopalinology is one of the classical techniques that is used to identify the botanical origin of the honey. We, of course, are used plant DNA that is contained in the pollen, and with the two target fragments of the plastidial DNA that is contained into the plants. And amplifying with these two regions, it is possible to obtain information that then will be sequenced. And after bioinformatic analysis, it is possible to assign the reads to the corresponding plants. So the process is quite simple. There is a DNA isolation from honey, PCR analysis using universal primers that target the so-called barcoding fragments that can be amplified from many different plant species using the same PCR primers, but of course the genetic information between the two primers is different and is species specific in many cases, or family specifics. Next generation sequencing can be used to obtain this information, and then there is the need to compare what is produced with a reference database that is purposely built to contain the information that is needed to translate what is produced by the sequencing approach. So we need to know in advance sequence that have a name, and based on that, using comparative approaches, we can assign the reads produced by this approach to the corresponding plant species. This is just an example. We have plenty of these examples. This is just an example from one honey samples that is acacia honey, and we obtained 1,000 of reads, short fragments, DNA fragment being sequenced, and this is the distribution of the number of reads in percentage assigned to the different species, genera, or families. So acacia honey, we assigned 41% of the reads to Robinia. Robinia pseudoacacia is the species, the acacia species. And we assigned also reads to many other botanical groups, more than 70 groups that could provide quite interesting fingerprinting, plant fingerprinting of the agricultural landscape where these acacia honey has been produced. Actually, this sample came from Tuscany, and this is quite interesting approach. Of course, we can do this in many different ways for many different honey samples, and we did it for hundreds of samples. The other element is, okay, so the honey doesn't really contain only the plant DNA. It can contain also plant sucking insect DNA. Plant sucking insect can produce honeydews, that is another alternative source of sugars for the honey, in addition to nectar. And we demonstrated that almost all honey samples contain an entomological fingerprinting derived from honeydews, even if the honey samples are not declared to be derived from honeydews. We produced this paper recently, and where we analyzed the insect metabarcoding, trying to get information from the insect that produced the honeydews that was used by the honey to produce by the honeybees to produce the honey. Here we target mitochondrial DNA using a universal primer that we designed for the Hemiptera group of insects. So the Hemiptera metabarcoding is similar to the plant metabarcoding approach that we designed, and I explained it before. And the process is similar: DNA isolation, PCR analysis with universal primers that target all insects for this specific mitochondrial DNA region, next generation sequencing, bioinformatic analysis, and of course we need also to construct a very specific database that can interpret the information obtained in this way. And these are just an example of what we produced. We analyzed several honey samples. Some of them came from honey dew honeys. Were labeled as honeydew honeys, and most of them include the reeds that came only from Metcalfa. The Metcalfa is quite common insect that produce honeydews, and this honey contained only Metcalfa reeds, and other honey samples contained other type of plant-sucking insects. For example, we analyzed the silver fir honeydew coming from Emilia-Romagna, north of Italy, and another sample, silver fir honeydew, derived from Piedmont region, again in north of Italy, and these honey samples contained only reeds from Cynara pectinate. Cynara pectinate is an aphid that is very specific for the silver fir. So there is an indirect way to identify the botanical origin of this type of honey that is derived from the honeydews that is contained into the honey. The other silver fir honeydew honey contained not only reeds from Cynara pectinate, the green part of this graphic, but it contained also Metcalfa reeds, and also contained many other insects that were here identified coming from different species. So there is a quite interesting entomological fingerprinting of the honey that can be captured in this way. This is another honey samples, apple tree honey, that contained reeds from Metcalfa, and also many other reeds derived from many different species. Here there is the largest number of other reeds came from Ropa Lopsifum incertum, which is called also apple grass aphid. That means just the name can confirm that there is a direct involvement of this aphid into the ecology of the apple tree. So indirect information coming from honeydews again to give a fingerprinting that could then attribute it to the apple tree honey. Okay, so let's move quickly to other elements. Genetic diversity of the honey bees that could be also useful to combat frauds, but also to monitor the genetic diversity of honeybee populations. We can target mitochondrial DNA and also nuclear genome DNA. So we published recently another paper where we authenticated using a endpoint PCR analysis. We obtained information on the potential subspecies derived from the mitochondrial DNA, Apis mellifera subspecies. Just a few words about what is Apis mellifera subspecies. Actually, there is classical classical classification in some way of the honeybee populations in Europe, in Africa, that divide honeybee subspecies into three main lineages. There is a lineage that is an African lineage. There is the M lineage that is Apis mellifera subspecies, and there is a C lineage that contains several other subspecies, Carnica, Ligustica, and other subspecies. So this is a very simplified classification of the subspecies based on mainly morphometrical analysis. Morphometric analysis is the classical way in which Rutner, the pioneering work of Rutner published in a book, classified the different Apis mellifera subspecies based on many parameters that go from angles of many different parts of the body of the honeybees. Of course, we cannot just use honeybee information to identify the subspecies that produce the honey directly from morphometrical analysis, because of course the honey cannot be directly analyzed for morphometrical parameter, but it can be indirectly using the DNA. There are three main mitochondrial lineage in Europe that pair with morphometrical classification, A, C, and M. There is a partial overlapping of the mitochondrial lineages with the morphometrical classification, and it is possible to use endpoint PCR to identify the three main lineages based on some differences in the amplified fragment that can be obtained from mitochondrial DNA. Actually, the A lineage has a long fragment, the M lineage is an intermediate fragment, and the C lineage has the shorter fragment if we target this mitochondrial DNA region that is the COX-1, COX-2 intergenic spacer that is called the tRNLA gene, and so on. Okay, so the analysis is quite simple because it is a PCR and then a fragment analysis in a classical agarose gel electrophoresis. It is possible to distinguish very easily the three main lineages. Of course, these are only samples that have been amplified with this primer pair, and some only samples can contain multiple bands because, of course, it could be produced by more than one colony, where there could be more queen honeybees that could carry different mitochondrial DNA lineages. This analysis make us possible to authenticate indirectly the origin of some honey samples because in Sicily there is the Apis mellifera siciliana, which is considered from the A lineages, and all honey samples that we analyzed that have been declared to be produced from honeybees of the Apis mellifera siciliana contained only the A lineages in the mitochondrial DNA. Other honey samples that we purchased from the Iberian Peninsula contained the A and M lineages. That is because it is, of course, the two lineages may be present due to population genetic aspect of the mitochondrial DNA genetic information from this region. But also we identify one honey samples that contain only the C lineages. So this probably is a honey sample that could not be assigned to what was declared to come from the Iberian Peninsula. Only this it raised our suspect, of course. Then we used this approach to obtain a population genetic information in Italy derived from the distribution of the different mitochondrial DNA lineages in Italy. We analyzed more than 800 samples, honey samples produced in 2018. Throughout Italy, and of course the dimension of these graphs indicate the number of samples that we analyzed for the different regions, Italian regions, and the color indicate the type of mitochondrial DNA profile that we identified from the honey sample that we analyzed coming from these regions. If there is just a red part of this graph, only the A lineage was identified. The green part indicate only the C lineage, and other colors indicate that honey samples contained 2 or 3 different lineages. It is interesting to see that actually there is quite uniform distribution of the C lineages, only sample that contained only C lineage, but also other only sample that contained the A lineages. And this is quite worrying compared to previous pictures that have been produced over the last years. So this is distribution of the different samples that we obtained. A sample that contained also A lineages indicate that there was introgression of the A lineages throughout the populations of the whole Italian peninsula. This is probably derived from the use of hybrid queen that were introduced by the beekeepers in different regions, because they are not originally considered to be original from the Italian peninsula. And this is the comparison that we can provide. This is the picture that we obtained in 2018. Actually from the honey samples that have been produced in 2018. And this is the picture that was available from a publication was published in 2000 using not only DNA, but using mitochondrial DNA analysis directly from the honeybees. And the different colors here indicate that there was a C lineage, white, and the M lineage, the M7 lineage in different parts of Italy, and the C lineages is the white circle, and the black lineages is the M4. The black part of the graph indicates the M4 lineages that is coming from the Apis mellifera. And also there is part, there should be some part of the Italy in which at the border there is a contact region between Apis mellifera ligustica and Apis mellifera carnica that was not really described here. So what is clear, there is a completely different situation of the mitochondrial DNA population genetic information from Italy over the last 20 years, because we have many A mitotypes covering all over Italy, and also we have a decrease of the M mitotypes in many parts of Italy. We also went into more details to analyze not only the three main mitochondrial DNA lineages, A, C, and M, but also we were able to distinguish C1 from C2. C2 is the classical mitochondrial lineages of the Apis mellifera carnica subspecies, even if there is discussion on this question. And based on another assay, we were able to discriminate the presence and distinguish the presence in the honey of the mitochondrial DNA coming from these two subspecies, Apis mellifera ligustica and Apis mellifera carnica. All right, so this is just another application that we produced using honey bee DNA that is contained in the honey. And can be also interesting for monitoring the inbreeding level of the colonies. We targeted the complementary sex determiner gene, CSD gene, because this is a gene that is involved in sex determination in Hymenoptera. Actually, we have to explain a little bit the question here. Apis mellifera sex determination is based on haplodiploidism. So the drones are haploid, and the queen and the worker bees are diploids. And the haplodiploid means that the males are haploid and females are diploids. Actually, this is not completely true because this is related to the heterozygosity level of CSD alleles. So females being diploids contain 2 alleles at this gene, and these 2 alleles should be different to obtain females. If there is homozygosity at this gene, so the 2 alleles are identical, there is a production of diploid males that are eliminated from the colony because they are not vital and they are recognized, so they are eliminated from the brood, and the brood appear, if there is a high level of inbreeding, the brood appear with many holes into the brood. And the males are hemizygous, so they contain just 1 copy of the alleles. In the population, there could be many different alleles segregating, and what is important is to maintain a quite high level of diversity within the population of this allele. This gene is very interesting. It is composed by 9 exons. There is a very important region that is a hypervariable region where there is a high level of variability. There are many different sequences that are produced. There is a very interesting mechanism that maintain high level of variability. But what is important is this level of variability is derived by the different in the amino acid sequence and the length of the amino acid part of the protein in this region. Here again, we wanted to analyze the level of variability of this gene, or this fragment of gene into the honey. And the same approach we used: isolation, DNA, PCR with primers, specific primers that flanked the hypervariable regions, next generation sequencing, bioinformatic analysis, identification of the CSD alleles, and then database. We constructed database to compare the sequence that have been already described before by other studies. And what we obtained, I would skip this. I quickly move on this. We obtained a quite interesting picture of the alleles that were present in different honey samples. Actually, this honey sample that we analyzed came from honeycombs, directly sampled from a colony. So they are not just on the bottle, so they might be derived from just 1 colony. And the province, the number of reads, the number of retainer reads, the number of CSD alleles in terms of protein alleles that we obtained. It's quite interesting because in some samples we obtained a quite large number of CSD alleles. Usually 1 colony should have from 10 to 20 CSD alleles that describe the number of drones that in some way were mated by the queen. So this is quite interesting picture because probably in some of these honey samples we sampled more than 1 queen, or maybe more than a fingerprinting derived from 1 colony, because it is clear that in some honeycombs there could be the contribution of more than 1 colony in many cases if this is derived by the beekeeper's practice. All right, I skipped this. This is the list of some of the measure alleles, CSD alleles that we identified in different honey samples. Honey samples are indicated with the H and the number of honey samples. There are the two main alleles that identify the alleles of the queen, but also there are major alleles that identify quite frequent alleles segregating in the population. Again, for population genetics analysis on the honey that target again the honey bee DNA, it is possible to analyze many SNPs from the nuclear genome, not only from just one gene, CSD, but covering the whole nuclear genome. And we developed the genotyping by sequencing approach that can disclose Apis mellifera population genomic information contained in the honey. And we were able to, based on this approach, were able to cluster some of the honey samples that in regions of the plot that could be in some way considered very close to Apis mellifera ligustica, because we also used SNP information from honeybees of the different subspecies, and this method can be also useful to authenticate the entomological origin of the honey based on the subspecies that produce it. Okay, health and pathogen using target analysis. We target many different pathogens, pest and parasite DNA that are quite common in apiculture, and we published this paper that is the first paper that we published for to monitor the level of pathogens from honey samples. Varroa destructor, this is just the endpoint PCR analysis. We analyzed more than 100 samples for many different pathogen, pests, and parasites. Varroa destructor were always detected in all samples. That was some way expected, and there was the presence of many different other pathogens or parasites or pests into the honey that we analyzed. We also then analyzed in more details the presence in north of Italy, based on the honey samples that have been produced in this part, in these regions of the Italy, the presence of a trypanosome parasite that is Loptaria passim, again using endpoint PCR, and where among the first two demonstrated that this parasite is quite spread into the apiaries in the north of Italy. We also developed a method to identify Echinna tumida from DNA contained in the honey, and this is the limit of detection that we analyzed. We obtained 0.0001 nanogram of Echinna tumida DNA that is present all over the DNA extracted from the honey, and we are able to identify among the samples that we analyzed one sample that was positive for Echinna tumida, and 60% of the samples were also positive for Galleria melonella. This is another interesting approach that we developed for Varroa destructor using metabarcoding, not just the targeted analysis. We sampled many different honey from different parts of the world. In blue, the position, the origin of the geographic origin of the honey sample that we sampled. Again, DNA was isolated. We designed universal primers, PCR primers that in some way target only Varroa and mitochondria DNA. We sequenced, and then we compared the obtained sequence with what is available in other publications, and we were able to assign the different lineages of the Varroa that was present in the different honey samples. The K1 is the most frequent lineages, the most aggressive Varroa lineages that is present in most of the samples. There were some samples from Brazil and Chile that contained the J1, that is classical from the South America. And also there is P1, that is a sub-lineages of the K1 that was also present from in samples that we collected in Colorado. There is also quite interesting result here, because in a sample that we purchased from China, we identified the C1 Varroa lineages that, as far as I know, has not been described to attach Apis mellifera, but only Apis cerana. So this is quite a worrying result that could raise some concern about another spillover from Apis cerana to Apis mellifera, but of course this is a matter of additional confirmation by other studies. On target analysis, finally. On target analysis, what we did, we took the DNA from the honey and we sequenced without any pre-filtering approaches. And this is a quite demanding computational analysis, because we need to translate the sequence information that we obtained from many different organisms into something. And we need to understand what are the species that are present. We first used an approach, a quite sparse method approach, because we wanted to reduce the computational burden of this approach with a few samples. And this is the distribution of the reads that have been assigned to different kingdoms. The green is the green part of the graph indicate that most of the reads, 93 in one sample, 67% in other samples, were assigned to viruses. The other most relevant fraction of the DNA with this approach was assigned to bacteria in both samples, and also there is a quite heterogeneous assignment to different kingdoms and type of organism, including arthropods. In blue, that means most of the arthropod DNA was assigned to ONIV, and to fungi and plants and other. So this is a quite interesting approach, because if we go into more details on the specific kingdoms, this is just a bacteria. We can identify many types of bacteria. I could obtain quite interesting fingerprinting of the hive ecological environment and where the honey has been produced. The other approach that we obtained is was based on Illumina sequencing with a high depth of sequencing. So the computational burden was quite heavy. And here we had to set a pipeline and then the corresponding database to translate the information that had been produced. We used this information for many different approaches and to extract relevant information. Again, all kingdoms were identified: invertebrates, mammals, rodents, phages, plant, fungi, bacteria, and many other have been identified with the prevalence of some of the bacteria and the virus and the honeybee DNA. If we then we wanted to investigate the honeybee DNA, and we were able to assign the subspecies of the honeybee that produced these honeys, just analyzing part of the genome, of the nuclear genome and the mitochondrial genome that was retrieved using a shotgun sequencing and not just a target sequencing approach. For virus was quite interesting, the analysis that we obtained in virus, because the most relevant virus was Apis mellifera filamentus virus. And for this is just analysis for 3 samples. One sample was produced in one apiary, apiary A, and 2 samples were produced on the same apiary, different colonies. So this is a FST analysis. So it is a population genetic approach that identify the want to identify the difference between the genetic difference between populations, and if there is a high level of FST, it means there is a quite large genetic diversity between the organism. So there is a quite large diversity between the Apis mellifera filamentus virus coming from the 2 different apiaries, but not from the same apiaries, different colonies. So this is a quite interesting result that we obtained. In conclusions, honey environmental DNA provides information that can be used for many different applications. This is clear. And honey environmental DNA opens an amazing window on a multi-kingdom derived fingerprint that describes the honeybee or genome complexity in many different parts. I would like to acknowledge the founders of our research, Regione Emilia-Romagna, the Minister of Agriculture, Italian Minister of Agriculture, and we have another project, Itaoni DNA, and I would like to acknowledge many breeders organizations, beekeepers organizations, Lucia Piana, and the Osservatorio Nazionale Miele, UNAPI, FAI, and Miele in Cooperativa. And also I would like to thank again all my collaborators who did a great job to produce these results. And thank you for your attention.
Thank you a lot, Professor Luca, for this awesome presentation. Do we have any questions? We have something from Violetta.
Hi, can you hear me?
Yes.
So excellent presentations. Thanks to the Dr. Luca Fontanesi. This is like a natural history of all that we have to know about honeybees until now. So thanks for the group and everything that you've done. In these days, like the last years, I trying to combat like some beekeeping and veterinary practices together. And one of my point is to find genes of antimicrobial resistance in honey, because when we eat honey, we also eat some spores from some bacteria that are very well known, but no one take care about that. What I want to suggest you and to share with the community here is the big need to do some survey on the samples that you already have concerning the presence of the genes for antimicrobial resistance for the different kind of antibiotics that are used and abused during the practices. Maybe not in Italy, but in many parts of the world, this is like a hidden thing that the beekeepers using the big amount of antibiotics that can change the structure of the genomes of the bacteria that are against use. Just your opinion and the hope that in your samples you will be able to trace those findings, because they are very valuable in these days. And thank you for the too long question and comment.
Yeah, I can comment on that. Of course, the use of antimicrobial is forbidden in many countries, not in all, unfortunately. And this is something that we could monitor quite easily if we analyze the DNA contained in the honey. Actually, the shotgun sequencing can produce many different type of information, and we wanted also to enter into this analysis, of course, but we started with a very other very interesting questions. But of course, this is another interesting question that could be in some way targeted, especially using shotgun sequencing, in my opinion, because in this way you could get many different sorts of resistance in some way. Of course, there is a matter of setting up a proper bioinformatic platform to recover this information.
Thank you.
Thank you.
We have a few comments in the chat from Anna Muti. For first one is, why do you use Streamland genes and not ITS2? Second one is, maybe we can go first one by one.
Yeah, okay. Yeah, well, actually, this is just what we published. Thus far, we are in the pipeline many other meta-barcoding approaches, and we are testing many different mitochondrial and plastidial systems of barcoding. Some of them are quite effective, others are less effective, depending on the honey degradation, because this is relevant. You need to identify a quite informative short meta-barcoding target, because if you want to be sure to amplify all honey samples that could be available, you need to have the possibility to distinguish botanical information using short fragments. And this is not obvious for honey, because depending on how honey are treated, the DNA is highly degraded, and in many cases it's not possible to amplify longer fragments.
Oh, okay. Do you use a homemade database created from the samples that you have, or NCBI information?
We obtain a combination of information. We use the combination of information.
Mm-hmm. Okay, nice. Can you reach a species level for plants?
Yes, in some cases, yes. In other cases, no, depending on the type of barcoding and the type of honey. Depending on how informative is the barcoding and the barcoding system compared to the different families or subfamilies or something like that.
Mm-hmm. Okay.
In some cases, yes. In some cases, we reach a very high level of informativity, and also we have other where we do not have the possibility to distinguish different species, and it is very relevant to distinguish between species. We have set up very specific target assays that will be published soon.
Sorry, Jankovic. I want to ask a question related to this issue. Do you make the pollen analysis in honey, and there's any correlation between DNA analysis and pollen morphological analysis of honey sample?
Yes, we have melissopallinology information from most of our honey samples, and there is, depending on the type of honey, there is a good correlation. Good correlation doesn't mean statistically speaking, because using metabarcoding, you get a lot of information that is not obtained using melissopalynology for many reasons. So you can usually get many plant groups, a much larger number of plant groups using metabarcoding approaches than using melissopalynology. That is clear.
Thank you very much.
Thank you.
Okay, we have two more questions regarding insects metabarcoding. Maybe you answered them already. Did you block Apis mellifera DNA, or did you remove it from the results after?
We designed the primers to reduce the possibility to amplify honey bee DNA, but of course we filter out after the sequencing to be sure that we got very specific DNA information using universal primers. Sometimes is very difficult to have only the amplification of other plant-sucking insect apart from honeybees.
Okay, and one more, which I'm most interested about. How can you use Apis mellifera genetics to determine honey fraud? Geographical provenance?
Yes, well, actually, there are in some honey, there is the need to label to indicate the subspecies of origin of the honey. And this is related to PDO product in many cases. There are some PDO products in the Iberian Peninsula and in other parts of Europe. And based on that, there is possibility to identify indirectly the geographic origin of the honey. Of course, this is indirect information. If there is a clear indication of the subspecies on the PDO production systems, of course, you need to rely on the definition of subspecies. And then use DNA information that could also useful to identify the subspecies and to discriminate these subspecies.
Okay, thanks. Anyone else got some questions? If nobody asks now, then I suppose we are done. Thank you again, Professor Luca, for giving this awesome presentation.
Thank you. Thank you very much.
See you all next month for the next webinar. Bye-bye.
All right. Very good.
Bye.
Thank you. Bye-bye.